I'd like to thank the organizers for inviting me to give this talk. I'm going to start off the talk with this simple cartoon. And really the point I want to make is that diet in particular can change the microbiota composition. And the microbiota composition can affect dysbiosis and the development of all of these diseases you see on the right. So I think it's a very straightforward concept that diet, particularly Western diet, might be driving all of these diseases and represents, I think, a much better explanation for things like asthma, allergies, autoimmune diseases than the prevailing hygiene hypothesis. So I think diet and the microbiota, I think, could be a unifying mechanism for all of these major burdens of human health shown on the right-hand side. So let me tell you how we got into this really exciting field. We kind of got into it from the other end. We discovered a receptor that then, when we worked back, implicated the microbiome. So GPR43, we fell upon this through various transcriptomics analyses. There's a really interesting receptor. And in fact, when we went to the literature, GPR43 was shown to bind short-chain fatty acids, a product of the gut microbiota. And in fact, short-chain fatty acids were previously reported to display anti-inflammatory effects, but this was largely ignored, 20, 30-year-old data. The fact that dietary fiber, which yield short-chain fatty acids, was also shown to be anti-inflammatory, also largely ignored. And even the effects of probiotic was very controversial. And probiotic bacteria produce large amounts of short-chain fatty acids. In the bottom of this slide, I've shown a cartoon, which is basically the model, that fiber is digested in the gut by gut microbiota, which have the enzymes to digest fiber. We don't. Short-chain fatty acids are produced in enormous quantities, and there's a receptor expressed on immune cells and on gut epithelium called GPR43. So it's hard to imagine that this receptor isn't doing something really interesting, and that was in fact what we found. When we did the DSS colitis model, on the left, this is the normal destruction of the epithelium and the inflammation. And on the right, in the 43 knockouts, we got this much worse inflammation and much worse severity of disease. When we put short-chain fatty acids into a normal mouse, we improved their disease, but not in the 43 knockouts. When we just looked at germ-free mice, which don't have short-chain fatty acids in high amounts, we got something similar to the right. It's almost like the ligand knockout rather than the receptor knockout. So all of this was really implicating this receptor and the short-chain fatty acids as incredibly important mechanisms in the pathogenesis of colitis. So if short-chain fatty acids are doing these great things, you'd expect fiber to do the same, because fiber is broken down to short-chain fatty acids. We fed mice a very high fiber diet and got a really remarkable degree of protection in the DSS colitis model. We did the opposite. We fed mice a low fiber or zero fiber diet and got horrible disease in these mice, the destruction of the epithelium and the worse inflammation. And in this paper, we showed that these mechanisms were relating to activation of the inflammasome, a pathway that had previously been implicated. In this paper, we showed that the mechanism for fiber and short-chain fatty acids was through activation of the inflammasome, a pathway that had previously been implicated in the epithelial integrity in DSS colitis. Up on the right, it wasn't just inflammation, it was also cancer. Low fiber gave us more cancers in the colon and bigger cancers. High fiber protected. And this is somewhat reminiscent of the observations of Burkitt in Africa, who observed that Africans don't get colon cancer, whereas Europeans do. And he hypothesized that it was the high consumption of fiber in Africa that protected them from cancer. We looked at the microbiome changes, and like everybody else, we see things go up and things go down. In fact, mouse chow is reasonably high fiber. And so giving a very high fiber diet changed it somewhat, but the most dramatic change is when we fed the low fiber diet. And this is, of course, is that there's different species of bacteria that will expand if they've got a substrate like fiber or not. And it's these species of bacteria in zero fiber that expanded that gave us such horrible DSS colitis. And conversely, the species that expand in the very high fiber are extremely protective in colitis, but also many other diseases. So I'm going to go through now a whole lot of different models of different diseases and just show you how beneficial short-chain fatty acids and dietary fiber is, at least in the models. And I'm going to start with food allergy. This food allergy is a disease that barely did not exist, when I was a boy, 40, 50 years ago. I didn't know anyone at school who had food allergy. And you can replicate food allergy very well in a mouse. You can sensitize, in this case, to peanut extract and then challenge and get anaphylaxis. And if you just look at the bottom left-hand side here, anaphylaxis is really much higher in zero fiber fed mice versus high fiber fed mice. The same with IgE production, one of the main mechanisms of food allergy. And when we look at the knockout mice, 43 knockout and another receptor 109, you see higher anaphylaxis scores in these receptor knockout mice, really indicating that what's going on in the gut and the tolerance and so forth is relating to these receptors. So one of the mechanisms that we uncovered in this study was when we fed zero fiber, we didn't get very many of these CD103, tolerogenic dendritic cells. These were, greatly expanded in the high fiber fed mice, at least in the mesenteric lymph nodes, but not with zero fiber. Now, these tolerogenic DCs are upstream of Tregs. They're considered the master regulators of tolerance in the gut. And when we looked at Tregs in the gut, using a FOXP3 GFP reporter, you can see just with acetate feeding, or fiber not shown here, that the expansion of these cells, they just lit up like a Christmas tree when we fed fiber or acetate, and on the right is shown no acetate. And of course, these Tregs are important for immune tolerance in the gut to things like food antigens, even bacteria. Unfortunately, we were beaten to this story, and these are 3 papers, 1 in Science, 2 in Nature, on the role of short-chain fatty acids, particularly butyrate, in expanding Tregs or improving their function. And this is just a cartoon that just summarizes our study. It's, really we saw acetate going through GPR43 in one pathway, improving epithelial integrity and preventing the expression of upstream Th2 type cytokines like TSLP and IL-33, and this prevented the development of Th2 cells. Whereas butyrate was particularly good at binding GPR109 on these tolerogenic DCs and promoting Tregs. And so these 2 mechanisms together really helped prevent food allergy. And this fits with like an immunohistological expression of these receptors because it's GPR43 highly expressed on gut epithelium and GPR109 more expressed on dendritic cells in the lymph node, fitting with this model. So I'm going to go on now to a whole lot of other diseases quickly. And in fact, there's not just Th2 type diseases that have been going up, but also more traditional autoimmune diseases, multiple sclerosis, type 1 diabetes, Crohn's disease, and so forth with Western lifestyle. And so we decided to look at type 1 diabetes, and we got this, I think, quite amazing result, or we replicated an amazing result by Chavonsky. And what Chavonsky and what we confirmed was that when you take NOD mice and you cross them to the MYD88 knockout mice, these mice, when you make them, when you look at incidence of diabetes, they're completely protected. And I don't know why Chavonsky did this experiment, but he then made these mice germ-free, and then they suddenly got the normal incidence of diabetes. So a natural conclusion is there's something about bacteria in the normal mouse that is protective. And once it's germ-free and that's taken away, then they get disease. That was a conclusion of the paper. And so we looked at acetate and butyrate and so forth, showing here butyrate, but the same story for acetate. It's like screaming levels of acetate and, in this case, butyrate, particularly in the portal vein coming from the gut in these protected mice. So high levels of acetate and butyrate, really a smoking gun that this could be a mechanism for this high level of protection. So what we did was to test this, we used a technology developed in Australia called high amylose maize starch acetate, or high amylose maize starch butyrate, where butyrate or acetate have been added on through ester bonds to a resistant starch backbone. And then we fed mice this diet. And it wasn't just us who used this fantastic diet. It's been published in Nature a couple of times and by us in Nature Immunology. And you can really get beautiful levels of acetate in the feces or in the blood with like the acetate diet, or butyrate with the butyrate diet. It's a fantastic way of delivering to the gut and beyond very high levels of short-chain fatty acids. And we could see this in the portal vein. We could get fantastic levels of acetate and butyrate, similar to the levels that we saw in those MYD88 NOD mice. And so what did this do to the model? We got almost or virtually complete protection from diabetes with the acetate and butyrate fed, like a mixture of acetate and butyrate diets. This is just a dietary supplement fed to the mice, protected them. And this was delivered after tolerance had broken down, which is a good sign for human patients that present with type 1 diabetes. Acetate alone or butyrate alone was still protective, but not as much as the combination. And I haven't got time to go through all the mechanisms, but one of the beautiful things I think about this type of approach, this dietary approach, is that you can hit so many different mechanisms. Not like a single drug that's hitting a single receptor. You can do so many things with a diet. For instance, we improved gut homeostasis and the expression of IL-22 and the suppression of inflammatory cytokines. We increased Treg biology, which of course is important for tolerance. We had effects on autoimmune, affected T cell numbers, which were greatly reduced. And we also changed the gut microbiota, which in a beneficial way, which also had an impact on disease incidence. And I'd like to acknowledge all the people listed at the top here. So let me move on to something else, which is asthma. And I want to demonstrate a different angle in this study. So we showed that just giving high fiber diet was beneficial in asthma models. But then we had an idea that maybe there's something going on even before birth. So what we did was we fed either high fiber or acetate to pregnant mice before birth. And then when these pups were born, they were transferred to other mothers to exclude a microbiota effect from the mother. And then they were just, as adults, then put through the asthma model. And so the only thing that these offspring have had done to them is an effect in utero with either acetate or high fiber of the mother. And then we got, and shown on the bottom here, eosinophils in the bowel fluid greatly reduced, particularly in the acetate fed, but also in the high fiber fed mice. And these mice have only had an effect in utero. If you just look at Th2 cytokines like IL-5 or IL-30, and the same story, greatly reduced levels of these cytokines just because their mothers were fed these things while they were in utero. If you did the lung function test, you couldn't even get a lung function test with these pups whose mothers were fed acetate or high fiber. So it really speaks to this whole notion of a developmental origin of disease, where influences in utero or in early life somehow set you up in later life for disease or not. And this is an example of asthma. We're getting exactly the same results in cardiovascular disease and Alzheimer's disease and others. So I'd just like to relate everything we've seen with asthma with some of the old theories. Junk food, the epidemiology of asthma and allergies. Junk food associated with asthma. Well, that's low in fiber. Mediterranean diet protects, that's high in fiber. Growing up on a farm protects. Well, you're exposed to livestock feces and short-chain fatty acid producing bacteria, so that could be protective. Antibiotic use might wipe out your good bacteria that are good at digesting fiber. Growing up with a dog, that's a little bit more difficult, but maybe there's a role for the aryl hydrocarbon receptor pathway and indoles that are also protective. So this is a bit now tongue-in-cheek, but I'd like you to consider what are the most prevalent immune diseases, and apologies to Family Feud. And you might be thinking, well, rheumatoid arthritis or lupus, and think, no. Some of these diseases, which were never considered immunological, are having a major immunological basis. Cardiovascular disease and hypertension look to be majorly inflammatory. Type 2 diabetes, Alzheimer's looks to be totally immunologically based, as is fatty liver. And so it may be that all of these major health burdens have some sort of connection with the microbiota, with short-chain fatty acids, that is very much immunologically based. And this is all very much tongue-in-cheek. These numbers are just made up here. Let's start with hypertension. I'll just briefly mention this study from Francine Marks, who published this using the DOCA model. Blue on the left is signs of fibrosis in high fiber fed mice or acetate fed mice. Beautiful protection in this model with these substances. And she later went on to show that receptors such as 43, 109 were involved. This is a study that Francine and myself and others published, where we used the DOCA hypertensive model, and this gives you nice fibrosis shown in blue on the left, and in fiber-fed or acetate-fed mice, we got this remarkable inhibition of fibrosis in this model shown on the right. And we showed that, in fact, GPR43 and 109 were responsible. I'm not going to labor all of this slide, maybe if you just look at the bottom right, the 43-109 double knockouts were significantly more fibrotic than the control mice. And all these other indices of hypertension and cardiovascular disease were also markedly altered in the 43 or 109 knockout mice, such as, diastolic blood pressure, pulse pressure, and so forth. Fatty liver, another major health concern. Really, really straightforward results. When we fed mice a low fiber diet, we exacerbated the ballooning and inflammation in this model. High fiber protected, but not in GPR43 knockout mice. Alzheimer's disease. One of the really interesting things about Alzheimer's disease is it looks to be particularly Western lifestyle associated. And as an example, individuals in the blue zone country of Ikaria in Greece, they consume a very high amount of fiber. And they don't get much Alzheimer's disease. And that's actually the same with many places around the world that are on a high fiber, non-Western lifestyle. So what we did was, we used the traditional Alzheimer mouse models, which aren't perfect, I have to admit. And the recognition of novel objects can be measured, and it's I think a reasonable sort of measure of cognition in mice, but it's the standard type of model. And when we did this in one particular model, the 5X FAD mouse model, the shown in green here is mice fed HAMS AB had a much better recognition index than either normal chow and particularly low fiber fed mice. And we've seen this consistently in every type of experiment we've done with Alzheimer mice. Whether we start the diet before birth or even after birth, which is promising because of course we're going to want to feed this diet to patients, once they show early signs of Alzheimer. And then when we looked at the knockout mice, we knocked out 43 and 41 in single mice and got worse disease in the receptor knockout mice. So how could short-chain fatty acids that are produced in the gut be influencing something like the brain? And look, anyone who's measured short-chain fatty acids will know that things like butyrate are virtually undetectable in peripheral blood. And that's in fact, what we're showing here. We used pigs here because we can then get at the small vessels from around the body. And there's no butyrate in peripheral blood, and there's very little propionate. And there's a bit more acetate, but not a huge amount. So then we started looking at some of these vessels coming from the gut, and particularly the small vessels, and then that feed into the portal vein. And we saw something very dramatic. And that is that you get screamingly high levels of acetate, of propionate, and even butyrate of around about the 1 millimolar at least in these small vessels. And an implication of this is that the blood that flows through this area of the gut and the cells in that blood will come into contact with these very high levels of short-chain fatty acids. And I would even suggest that this is a major mechanism of how the gut microbiome works. So and I'm calling this immunological tone. And so, for instance, short-chain fatty acids are transported from the gut lumen into the blood, and there's specific molecules that transport short-chain fatty acids. And so you have in these small vessels extremely high levels of short-chain fatty acids. And of course, there's immune cells circulating through the gut, as part of their normal circulation, that are contacting the short-chain fatty acids. And receptors like GPR43 flux immediately, within a second or less than a second upon, receptor, GPR43, for instance, engagement of these, short-chain fatty acids. So what I think might be happening is cells will be passing through the gut, maybe every 5 minutes, maybe every 10. It doesn't really matter. Maybe, every 20 minutes, but they're going to be hitting these high concentrations of short-chain fatty acid. And that's going to change their immunological tone. And then those cells will migrate to peripheral tissues, maybe the brain, maybe the liver, maybe virtually any tissue, and be influenced in their immune reactivity. For instance, their GPCR signaling will be altered in the blood. Maybe HDAC inhibition will come into play, or even AHR agonism. All of these factors will be majorly affected in these small vessels from the gut. So let me try and link all of these major human diseases with maybe, some common mechanisms. You've got things like inflammatory bowel disease and diabetes, cardiovascular disease, Alzheimer's, all have altered gut microbiota in humans, at least what has been reported in the literature. Now, when we have done mouse models for virtually all of these, we show an involvement of GPR43, the typical metabolite sensing receptors. And when we use high fiber diets, like the HAMS AB I mentioned, we get protection. In every case, we get protection from disease that we have studied. We haven't done atherosclerosis yet, or autism. So there's this really huge potential for very specialized, high fiber, high short-chain fatty acid releasing diets to be beneficial for human health if a human behaves similar to a mouse. So let me just show you one of the things that we're doing. We are very interested in inflammatory diseases. And so we thought maybe, we thought we don't want to just treat an inflammatory disease with a drug like a kinase inhibitor or a cytokine inhibitor. We want to cure the disease. And so our idea is maybe we can take out the offending subset, like a Th1 cell or Th17 cell, through depletion, then hit hard with these tolerance-inducing diets. And the two together might just lead to, complete remission. And this cartoon here just shows some of the subsets of immune cells, like Th17, which express CCR6 as a typical cytokine chemokine receptor, Th1 express CXCR3, and so forth. And so, as an example, here's mice treated with an isotype where you've got, these Th1 type memory T cells. This is depleted, almost every single cell depleted without depleting antibody. And so this would be like a great therapy for so many things, possibly IBD, rheumatoid arthritis, many. And then we would try and restore tolerance after we remove this offending subset with our diets. And so that's one of the major things I want to spend, I think, my career on at the moment, is to see if this is a possibility, this reset and restoration of tolerance approach, so we can cure a disease rather than just treat it. So this is the last slide, and herein, I think, lies much of Western lifestyle diseases, the mechanisms. What is it about our lifestyle, our Western lifestyle? Is it our Western diet? Is it, and how much is it antibiotic use? And so forth. What are the mechanisms? Is it, dysbiosis and leaky gut LPS distribution? But surely molecules like the short-chain fatty acids and perhaps many others, the aryl hydrocarbon receptor agonists, maybe even the longer-chain fatty acids like the omega-3 fatty acids, there's a deficiency of these. That's our model. And if you were to correct this deficiency and have high levels, then all of these diseases on the right may be prevented. And I think really we're beginning to understand the major mechanisms. On the bottom, certainly the G protein coupled receptors are not just the ones I've mentioned, but also many others that are metabolite sensing are having a major role. And then the other mechanisms I haven't mentioned, of course, is HDAC inhibition, which is more at a transcriptional level, and then transcription factors such as aryl hydrocarbon receptor. All of these mechanisms coming together, these are the three main ones, I think are really having an effect on this major health burden for human beings. And these are the people I'd like to acknowledge, which I won't labor. Thank you very much.